smell is not very strong. So I don't know.
All right. Is there anything up here that you have questions about? That you couldn't find, that you don't remember, that you just want me to explain again?
The last one.
This compare and contrast. So embryonic stem cells are the ones that exist kind of in that second phase of development, when the only differentiation that's happened is some of the cells have turned into the placenta. But that inner mass of cells, those are called embryonic stem cells, because they still have the potential. They're pluripotent. They can still turn into any kind of cell in the organism. The only thing they can't do is become part of the placenta. And so embryonic stem cells, if you take them at that point and put them in a petri dish, they will continue to divide and divide and divide. And if they're given different environmental cues, they can turn into any cell type. Adult stem cells are the ones that are still in your body now. They are multipotent, which means they can still turn into multiple types of cells, but not as many. Like the stem cells in your bone marrow can only turn into bone and blood cells. They can't turn into muscle or brain or skin. But you have different stem cells. They're just a little more limited. So that's the difference. Other questions? Okay. Today we just have a short introduction to wrap up what we were talking about yesterday. So we are going to finish the last section of the Unit 8 notes today, and connect this idea of development back to the idea of cancer, which we talked about earlier in the unit. It's possible the last couple slides I put up today, I honestly don't remember where I stopped printing these slides for you, but I know I cut off some of the ones at the very end about cancer. So if you don't have the last one or two, that's okay. It's actually information that we've already kind of talked about, and it's information that you are going to be looking at after this brief intro. You're going to be going to a website and doing a click and learn activity to look more specifically at one gene that's really commonly mutated in a lot of different cancer types. So if there are some notes at the end here that I didn't write or didn't print for you, don't feel like you have to write absolutely everything down. Some of it's review of what we've done before, and some of it you're going to go and be reviewing on your own in a minute, okay? So this slide was up there yesterday. I probably didn't reprint it for you today, but this is just so you kind of remember where we were and what we were talking about. We were at those earliest stages of development. And yesterday we talked about how within those first few weeks, there are kind of three main processes that help a zygote, a fertilized egg, become the organism that it's going to be. There, of course, has to be a lot of cell division. And that happens differently in different parts of the body. That's part of what helps it to grow and then eventually to have the shape of the adult organism. Yesterday, we focused mainly on the process of cell differentiation, the signals that those cells receive, some of which are already in the zygote, in that fertilized egg, because they were in the cytoplasm of the egg cell itself. Those are kind of the first signals that start turning genes on and off as that cell starts to divide. And then those cells, as soon as there's a little ball of cells, start sending signals to each other, telling each other what to do and which genes to turn on and off. And that's what starts differentiating those cells into different tissue types. The third process that has to happen that we're going to talk briefly about today is called morphogenesis. Morpho refers to shape. So that these are the parts that kind of take us from a blob, a circular blob of cells, to an organism that has a body shape, that has a head and a tail and limbs and a back and all that kind of stuff. So that's what we're going to talk about a little bit at the beginning here. And then I'm just going to make some connections so you can kind of understand how cancer fits into some of these things we've been talking about. One really important process in morphogenesis, in determining the shape of a finished organism, is apoptosis, which we've referred to a couple times. It's programmed cell death that happens because of cues that those cells receive that basically tell that cell to self-destruct. Apoptosis plays a really important role in development. So it does also play a role in mature organisms. This last point on here, cells that are infected or damaged will be triggered to go into apoptosis so they don't pass on that damage. And this is why apoptosis is related to cancer. One of the things that can contribute to cells becoming cancerous is if they don't go into apoptosis when they normally would. Because usually cells that have damaged DNA would get cues to, instead of continuing the cell cycle and dividing and passing on that damage, they would get those cues to self-destruct and go to apoptosis. So if they don't, that can be one of those things. But apoptosis is also important in development. The way that this happens, it's a very structured, regulated, organized process. It's not like the cell just implodes. The DNA is broken down. There are specific enzymes and chemicals that are made that break down the DNA and the organelles and the other things within the cytoplasm. The cell starts becoming really multi-lobed and blobby looking, like that one on the right, as its contents are kind of packaged into vesicles, which are then later engulfed by scavenger cells and broken down. So it's a very structured, regulated way to get rid of cells that are damaged or cells that aren't necessary when you're trying to make certain body structures. So here's how we see this in development. This is very important in the process of nervous system development. You might have heard, if you know anything about how kind of how the brain grows throughout your life, you may have heard that there are times when your brain's growing really, really fast and making lots of new connections. And there are other phases in your life when your brain is kind of pruning out the pathways that haven't been used as often. Apoptosis plays a role in that process. It also plays a role in development, specifically in determining the shape of an organism. And one of the easiest ways to think about this is the development of things like hands that have fingers. The same happens with paws and other types of extremities that have a very unique shape. This isn't the only place it happens. It's just a really easy way to observe this. When that part of an organism starts to develop, like most other parts, it happens through cell division. And so your hand actually begins to form as just a big blob of cells, kind of like the picture up on the left. But then the cells that are in the spaces that will eventually be in between where your fingers are receive signals to go into apoptosis. And so in this picture in the upper right, all these cells that are kind of bright yellow in there, obviously in these pictures were tagged with some chemical that made them visible. They are going through the process of apoptosis. And so all of these cells go through that process and kind of self-destruct, which creates spaces between your fingers. So that's just one simple example of why this is important in development and how it helps determine the shape of organisms. Questions about that? The other thing that's important to determining the shape of organisms is setting up the basic plan of the body. Which side's going to be the head and the tail, the back and the front, where the limbs going to be, all that kind of stuff. And this is sometimes referred to as pattern formation, and it's also controlled by molecular cues. So these are signals that cells send to different parts so that they kind of know their location relative to each other and where they are in that blob of cells. There are a few things that can control this. At the beginning, it's those cytoplasmic determinants that come from the egg cell, those molecules that are already in the egg cell. Like that picture we saw yesterday, where as soon as that zygote divided, the cell on the left had more of the red signaling molecules and the one on the right had more of the green triangle molecules. That already helps those two cells know that one is on one side and one is on the other. That's the beginning of this positional information. Other signals, as those embryonic cells start to signal to each other, those signals also help determine where the back and the front is. And there's also a set of genes, you might remember hearing in 10th grade about Hox genes, that are specifically in charge of setting up the body plan. Hox genes are like master switches in the genome that turn on and off other sets of genes to determine where the head is and where the wings are and where the limbs are and where the tail is. And Hox genes are very similar amongst many, many organisms. Like any organism that has 4 limbs, 2 arms, 2 legs, or 4 legs or whatever, has a very, very similar set of Hox genes. So these are genes that have been around for a very long time throughout evolution, and they've just been modified a little bit to change the body plans of different organisms. Any questions about that? So those things together, kind of this arrangement of the pattern of the body and apoptosis of the cells in between things, help to determine the shape and that process of morphogenesis. So now we're just going to make some connections and kind of bring us back. Like I said, if we had time at the end of this unit, I wanted to spend a little more time learning about some of the things that can cause cancer. All of the systems we've been talking about from that very first moment that an egg is fertilized, there's signaling within the cell and between the cells that tells those cells what to do and which parts of their DNA to turn on and off. And some of those signals control how those cells move through the cell cycle and which cells are supposed to divide more and which are not supposed to divide as they become those different body tissues. There are also parts of the DNA that control whether or not a cell goes into apoptosis. And like I said, that process is also helpful in preventing cancer and killing off those cells that might be damaged. So all of the same control mechanisms that we've been talking about that work throughout your life to regulate the cell cycle and determine how your cells become specialized and help those early development of an embryo, all of those same processes are the things that go wrong to cause cancer. All these notes are in Classroom also if I go too fast. So the important thing here is that the same systems that we were talking about in development are the things that go wrong during cancer. And this slide, you don't need to write these down because you're going to write this down again in the next activity. And we heard about this once when we watched that video last week about cancer. There are a couple main groups of genes that you often hear about associated with cancer. Proto-oncogenes are the ones that they liken to the gas pedal. They tell cells to move forward through the cell cycle. They say, yes, we're ready, go ahead and move to the next phase and start division. Tumor suppressor genes are like the brakes. They say, nope, we shouldn't divide, something's wrong, we're not ready, there's damage somewhere. Tumor suppressor genes can work in a couple different ways. They make proteins that might go and repair damaged DNA so that it gets fixed before that cell can move forward in the cell cycle. They might control whether a cell is attached to a surface. Remember, most types of cells need to be attached to something in order to divide. And so some of these proteins help with that process. And they might just tell the cell to stop and not move forward in the cell cycle. So there are lots of different proto-oncogenes and tumor suppressor genes. This isn't just one set of genes. And so we're going to talk about P53 in a minute, so I'm actually going to skip that one. And so because there are several different genes that control this, developing cancer is usually a multi-step process. One mutation is usually not enough to make a cell cancerous because there's all these backup systems. There's several proto-oncogenes, there's several tumor suppressor genes, and there are switches that turn all of those things on and off. And so usually one mutation isn't enough. You need to accumulate several, usually at least about half a dozen mutations in the same cell in order for a cell to become cancerous. This is the biggest reason actually that people tend to get cancer more frequently the older they get. Cancers tend to show up later in life because you accumulate those mutations throughout your life. And that's why, like, even when you're young, your parents hopefully bugged you to put on sunscreen all the time, because if you get sunburned too often when you're young, that can mutate your DNA once or twice. And then if you sunburn yourself again the next year, you could add another mutation. And so you're kind of starting to accumulate them more and more, which increases your chances of ending up with skin cancer when you're older. And usually it has to affect both of those types of genes. You have to break the gas pedal and the brakes in order for those cells to totally go out of whack. Like I said, there's lots of backup systems that your cells have to help prevent them from becoming cancerous. Questions about that? So today you're going to be looking a little more closely at one key gene called P53 that's kind of a master switch. It's a tumor suppressor gene, but it has a really important role, and it has been found to be mutated in a really high percentage of cancers. The activity we're doing today is officially part of your notes. I am not going to ask you to turn it in, and I am definitely not going to grade it because I have plenty of other things to grade before the end of the trimester. But this is important information, and this is a really common example of a cell cycle regulator that comes up in a lot of questions and situations. So on today's classroom post, I did not post this as homework since I'm not grading it, but on today's classroom post, there's a Google Doc that kind of leads you through what you should be taking notes on as you do this activity. That Google Doc also contains the link to this website. When you get to the website, click on that button that says Launch Interactive, and it'll start walking you through the click and learn. And the questions are very clearly labeled, kind of where in the click and learn you can find that information. If you want to type your answers into the Google Doc, that's fine. If you would rather write them in a notebook as part of your notes, that's fine too. I put the Google Doc there so you know what I expect you to know from this activity, but like I said, I am not going to grade that Google Doc. So if you'd rather put it in a notebook so it stays with your notes, that's really what this is. Does anybody have any questions about that? So you're going to have the rest of class today to work on this, and I think you should be able to get through it and finish this. This isn't meant to be homework, so hopefully you'll have this pretty much done by the end of class. People at home, you can go ahead and open that Google Doc for the link, and same thing, you can either complete the Google Doc or write notes somewhere else. That part is up to you. So you may sign off of the meet, and we will see you back tomorrow.
Can I turn this into your kid?